DNA methylation. In the gene expression, it was found that DNA methyltransferase, if this enzyme is activated, then DNA get methylation, and interestingly, the result is the gene expression was down. So this was the clue that maybe DNA methylation is the control of gene expression mechanism. Now, if you look at the structure of DNA RNA, now DNA has a ATCG, RNA has a AUCG. The difference is T has one methyl group here, U does not have it. Now it was found that the DNA methylation site was a cytosine. So this position here is similar to T. This site was methylated. If you look at the Watson and Crick base pairing, interestingly, this site is base pairing, and then methylation site is behind here. So this methylation of T comparing to U, that does not bother much of base pairing or DNA replications. So similar to this relationship, maybe if there is a C also has the room for the methylation. So this is exactly where the methylation on DNA happens. So this is a methylated C and this is a regular C. So in the base pairing, if you look at the DNA structure, there is major groove and minor groove, and the methylation site on C is behind of this the binding site. That's why the methylation itself does not bother with the recognition of a double strand. During the DNA replication, if the two strand becomes isolated, then leftover G will take up C, but that C is non-methylated. Then how can you know that the original cytosine was methylated or not? It is interesting to see not every C is methylated. There should be a sequence of C and G. And to pronounce this CG a little easier, we call this CPG. The P here is phosphate linkage. And this sequence is always a palindromic. So that means CG one forward side and it has a backward CG2. So in this case, if the two strand is separated, still there is a marker of a methylation left here. Utilizing this methylation pattern, we can figure out which site was methylated later. Then, can you guess how many methylation sites here? Can you calculate? How many are they? Now let's look at the gene expression again. So here is the gene, and in upstream there is promoter area which can recruit the transcription factors and help the gene expression. If you make the combination of a dinucleotide, it's a 16 combination. So statistically, the CPG is a 1 out of a 16. That's about the 6%. So on the right side, if it is scattered, then it should have like a 6% of a distribution evenly. But very interestingly, this CPG is highly niched in a special area, so-called promoter. And here, this ATG is a gene starting point. So from ATG, this is a sequence of starting and also amino acid methionine, the signal. From here, the new protein is synthesized. So promoter area and also very early part of the gene, there is highly enriched CPG part. That's why this area is called as CPG island. This observation implies that maybe this promoter area, the methylation pattern, is the key mechanism of gene expression. Now let's look at the DNA methylation effect. If the CPG is not methylated, gene transcription happens. So the DNA make mRNA. If the DNA methyltransferase methylate the CPG-C, then gene silencing happens. That means gene expression stopped. Now here, the other enzyme, TED enzyme, can remove that methyl group, then the gene can be turned on again. How can we identify the methylation site of a cytosine? There is chemical method, so-called bisulfite method. So in cytosine, if you add a bisulfite, that remove the amine group, and through this hydrolysis, it change this amine into oxygen. As a result, so cytosine becomes uracil at the end. Now, old C, if they are not methylated, they can be changed into U, but the methylated cytosine, they can remain as C. Now, unmethylated case, old C can be changed into U, then later by comparing those sequences. If there's any C that was originally methylated C, and then old C was changed into U, then we can figure out which one was originally C and also which one was methylated. So if you have interested gene, then we clone those genes and sequence first. Then it can give us the CPG site like this. And then by treat with bisulfite, then those unmethylated CPG is changed into UG. Then through the PCR amplifications, then we can change the U into T. Then by reading this, we can figure out which one is methylated with the black circle, and then which one is the unmethylated open circles. Another method is the utilizing PCR. In this case, this is a methylation-specific PCR. So we prepare methylation-specific primer, GCAA. If this sequence is not methylated, then the old C is changed into U, then the GCAA does not match. So there is no PCR product is generated. Now, if this sequence, the C is methylated, then that remains as a C, then this primer GCAA matches, then PCR product can be generated. So by reading this PCR product, yes or no, we can tell whether that position is methylated or not. So this method is very convenient and powerful if you know exact site where you want to see the methylation pattern. Not only for the one single position, if you need to know the global methylation pattern, you need a different technique, so-called methylated DNA immunoprecipitation. Here, the immunoprecipitation means utilizing antibody, you isolate your target sample. So this is your reference, the DNA, which has a partial methylation, and you break down by sonication, and then label this DNA with a red color. This is covering all the DNA in your sample. Those fragment of DNA is denatured, and then here you utilize antibody for methylation site, and you can selectively isolate only methylated DNA and label that with a green color. After you mix this sample together, and then through the DNA microarray technique, you can compare this, and red color show you those reference whole DNA sequence, and among them, the green color overlap together with the red color may generate the yellow color. They shows the position of methylation. Well, this example, not only using this kind of the hybridized the DNA chip, it can be directly injected into NGS. Then by sequencing, also it's possible to figure out the methylation site. So two different methods is possible. This technique can be used also for mRNA sequencing. In the transcription, when the mRNA is synthesized, this mRNA sequence is real genetic code. That's why we call this as a sense strand. This genetic code sense sequence matching to the amino acid sequence of protein. How about the template? This template is, in a sense, it's the opposite or the complementary sequence. That's why this sequence is only the act as a template. It does not have any sense. That's why we call this as an anti-sense. So by abbreviation, we call this strand as a cDNA. So cDNA and mRNA is a complementary sequence to each other. Now let's try to make the cDNA library. The first step is the mRNA extraction. So collect all the mRNA from one cell or one tissue. And we make this into the DNA. Usually DNA make RNA through transcription, but some virus can make the DNA from RNA. This is a reverse transcriptase, and this process is called as reverse transcription. In this case, the primer is the utilizing those poly A tail, then poly T can be the primer. And then this is a cDNA. And if this is a collection of all mRNA's cDNA, we call this cDNA library. Now here, mRNA is the more susceptible, more unstable than DNA. So selectively, we can remove mRNA by enzymatic cleavage. Now this collection of the cDNA library is ready to move to the next step. Now, utilizing this cDNA library, first of all, we amplify them by PCR. So forward, backward, we enrich those sequences, and then through in vitro transcription. So cell-free transcription, by adding the enzymes and all building blocks for RNA, we can make RNAs in vitro. During the process, we can insert biotin, which has a high binding affinity to streptavidin. And utilizing this, we also fabricate the DNA chip. So on this DNA chip, there is the reference single strand of the DNAs are prepared. And here the samples are added. And then fluorescence labeled the streptavidin is used to visualize those bound position of the DNAs. So by analyzing those DNA chip image, now we can see which gene is expressed and which gene is silenced. If you want to compare two samples, let's say one is from liver and one is from lung, then we can label those cDNA library with a different color, and then we can mix them together. And then we can compare which the protein or which gene is more expressed in liver with the red color, and which protein is more expressed in the lung in green color. That kind of a comparative analysis is also possible. Now let's summarize the DNA methylation map. So here is the reference gene. Now, through this methylation mapping, we can figure out which C is methylated. So original DNA sequence is ATCG quaternary system. Now we add up one more information, but this is also digital information. This is C, some C is unmethylated and some C is a methylated C. So this is maybe fifth nucleic acid in the DNA. So this is the result we can get from the DNA methylation mapping.